มันก็ขึ้นแล้วนะขึ้นได้โอเคขอบคุณมากน่าจะต้องเอฟจริงจริงกับเรา now it's already recorded right
Yes, I join.
thank you
Thank you
คุณ
ค่ะ
จะทำ I'm sorry, like it's almost a year, but I'm still not get used to Zoom anything. Oh, sorry. Today topic is mitochondria, about its importance in producing an energy for us. Life needs energy from food, you all know, and we use it. We change, we transform the food to energy. The word bioenergetics is a branch of biochemistry that focuses on how cells transform the energy from food to a very important molecule that which you already know, adenosine triphosphate or ATP, and use it. Today I will talk a little bit about ATP, how to use it, and the big part of this lecture is how cells transform energy and produce ATP. Okay. Let's see how food is processed to energy. This we already know. We eat food. The biomolecule like carbohydrate, lipid, and protein. And we digest it into smaller molecule and into the monomer, the smallest we can do, like glucose, fatty acid, and amino acid. And that food and that monomers will get into the cells. Mostly glucose, some of it can be fatty acid. Mostly glucose enter the cells and into the process called cellular respiration, which you already know anyway. And cellular respiration make DNA. Make ATP. And ATP, which is a molecule that has energy, will give the energy to new molecule, to make new molecule, and to or maybe a waste, and give it has a heat. So we know that this is bioenergetic. Food is transformed to chemical energy in ATP with cellular respiration. And for addition, the biggest part of the cellular respiration, very important one, is in the organelle that we are going to talk about today, mitochondria. Okay. First, to understand about energy, we will talk a little bit about ATP. Why we use ATP instead of using other kind of molecule, and how we use ATP. That's very small part of this one. I think you all already know it. ATP in your heart. ATP, adenosine triphosphate, is a molecule that look like this. Consists of 3 phosphate group, 1, 2, and 3. 3 phosphate group, 1 carbohydrate, 1 ribose, and 1 adenine. We call it adenosine triphosphate because it has 3 phosphate here. Now, look at their structure. You can see there is a small molecule that has a lot of negative charge here. 4 of them. Compared to the size, there has too much negative charge. Negative charge hate each other. They don't like each other. To keep it near each other, that means you have a very strong bond here, right? Very strong bond. Energy was in here, high energy. But because they really hate each other, the negative charge, so with a little bit of water, this bond can be break. And when it break, it release a lot of energy. Okay. So ATP is a molecule that capture chemical energy from the breakdown of food and release it to fuel to other cells, cellular reaction process. Okay. Another time, the energy in the bond is high, and when this bond break, the energy inside can be used to initiate other reaction. With a little bit of help, from water, the reaction called hydrolysis or lysis by water. ATP, which has 3 phosphate group, will break down and change into AD-P, only 2 phosphate, turn into AD-P. Okay. The energy that was released is 30.5 kilojoule per mole. And what will we do with this energy? This is how we use energy from ATP, or we call energy coupling. Imagine like a hill like this. If you want to pull a car right down the hill, it just come down spontaneously. You don't have to add any more energy to make this car come down. Sorry, somebody waiting to get in the meeting. Okay.
กด admit ได้เลยค่ะ
ตรงไหนเหรอคะแอมขอโทษกด
ตรง
admit
หน้าตรงกลางหน้าค่ะ
ตรงนี้เหรอคำว่า invite เหรอ ไม่ใช่
เข้ามาแล้ว
เข้ามาแล้วนี่เข้ามาแล้วนะใช่ไหมโมฮัมหมัดเข้ามาแล้วค่ะ ขอบคุณมากเลยที่มีนิสิตไทยอยู่สักคนหนึ่งช่วยอาจารย์หน่อยนะ
ได้ค่ะ หนูก็ไม่ค่อยคล่องเหมือนกัน
แต่ก็ตกลงเขาเข้ามาแล้วนะ คนเนี้ยโมฮัมหมัดอ่ะค่ะ
Yes. Sorry.
I'm sorry โมฮัมหมัด
Yes. No problem. No problem. I have problem in connection.
อ่าฮะ
Sorry.
อ่ะ but some reaction in our cells like this, like a car that is on the bottom and want to go up, they cannot go up by it cannot go up by itself. It needs some more energy to push this reaction up the hill. That's like the importance of ATP. Some reaction is exergonic. Sorry, is endergonic. Reaction need energy to put it up, to make it happen. So they need energy from exergonic reaction, which is ATP. Energy from ATP will drive this reaction up the hill. This is why ATP is in need for living. And energy in the reaction 1 and 2 will be coupled or add together. The energy coupling come from this word. For example, to make something, to make new molecule in our cells, it's non-spontaneous. It cannot happen by itself. It needs energy from ATP. This is an example. Like in the cell, we need glucose. In the cell, right? This is a cell. I'm sorry to draw like this in the cell, and cell need glucose as energy source. When glucose get into the cells, the cell didn't want it to get out, so it changed glucose with additional of phosphate group like this. So glucose will be difficult to get out. This is very important reaction. First step of glycolysis. In the reaction to add phosphate to glucose, need 14 kilojoule per mole of energy. They need energy. So we hydrolyze ATP. ATP will be hydrolyzed and spend 30.5 kilojoule per mole of energy. When we couple the two reactions, there will be enough energy from ATP to make this reaction happen. Okay, that's why ATP, that's how cell use ATP for survival. And this is the example of how cell use ATP. Active transport, sodium potassium pump in nervous system and in the brain. This is very known, most popular use of ATP. The other type of, another example for using ATP is the transportation of organelle, vesicle or organelle inside cell. There is a microtubule in the cell and there is a vehicle here, move organelle and or vesicle. Use ATP, sorry. Okay, wait for me. Okay, you very welcome. You can ask when you have a question. Okay. This one is important for you. I'm sure that some of you will work on how this will work on signal transduction, and this is the one of the most important mechanism of signal transduction using ATP. Sometimes some protein didn't work. It just sit there inactive in the cell. There will be an enzyme called kinase, MAP kinase, ATP, ADP kinase, something like that. Help wake them up. And that process need ATP to help wake it up. And the phosphate from ATP will add up in that molecule, and that molecule get activated. The other use of ATP is like the movement of flagellum. ATP will power the sliding of the microtubule in the flagellum, or moving of the cells. Later, I have the picture to show you the importance of the ATP and the movement of the cells. Some living and some animal has an advanced technique in using the ATP. In fireflies, there is bioluminescent, the light production in this insect. There is a protein called luciferin, and it was catalyzed by enzyme luciferase. With magnesium, ATP, and oxygen, it's turned out to be a light. There has many reaction, many kinds of reaction that needs ATP. Now you already finished the part of bioenergetic about the use of ATP. Now the other part of bioenergetic is how to produce ATP. And to study that, you need to study a little bit about the organelle that devote to ATP synthesis, which is mitochondria. Before this, mitochondria, you all know, mitochondria is definition of mitochondria is a powerhouse of the cells, right? And I think maybe 1930 something, they know about oxidative phosphorylation, know about Krebs cycle, and they thought that that's all that mitochondria is about. And the study about mitochondria turned into the study about origin of the mitochondria, about like 50 years to study about the origin of mitochondria. I think just until 1980, 1990s, mitochondria was looked into another aspect, not only about powerhouse of the cells, but in the induction of apoptosis or how to suicide the cell itself, which I will talk to you about the apoptosis later, very later, at the nearly final of this semester. So today, just focus on only about ATP, the things about ATP synthesis, and a little bit more. I don't know how to say it, but okay. Today, mitochondria, we'll talk about 3 topics: its structure and functions. Okay, like a foundation for you, and this is the important things: oxidation and ATP production. And because of you are a veterinarian, we come here to study about disease, how to protect, prevent, or treatment. We talk a little bit about mitochondrial diseases. In veterinarian, it's also, I think it can be applied into many topics, and it's still like a new aspect for veterinarian. Mitochondria is a membrane-bound organelle. There is 2 layers of membrane. Mitochondria produce ATP. Its name, like you already know, the powerhouse of the cells. Mitochondria is special. They have their own small chromosome, mitochondrial DNA, or you can say mitochondrial genome. Which has different, some of it has a different code from when you are different codon from normal. And generally, mitochondria and mitochondrial DNA are inherited only from mother side. It's come from your mother. Your mitochondria come from your mother. Structure and function of mitochondria. It can be found. This is, I don't know this, the red one is, but it usually it should be cytoskeleton. The blue one is nucleus. And the green here, it's mitochondria. This is a MDCK cell. And this one I would like to show you. This is the cell of osteoblastoma cell. This one is actin. The purple one is actin, cytoskeleton. This is nucleus. And this cell is moving. Moving need energy for actin to move. And you can see the relationship between actin and mitochondria. The relationship between moving actin and mitochondria. The mitochondria was pulled into only this part, which need energy to move. No mitochondria here or here. You can see the function of mitochondria clearly about making energy for cell to do a thing. And this is electron microscope of mitochondria. This is nucleus. And this is mitochondria. You can see the outer part, outer membrane part, and inner membrane here, like this. The brown one, sorry. This brown one, outer membrane. The blue one is inner membrane. Inner membrane is a lot bigger than outer membrane. It's bigger, but it has to be inside. So there is a fold inside. It's called cristae. Cristae or cristae, you can say both. In liver cells, the inner membrane is 5 times bigger than outer membrane. But in some cells that is heavily using energy, like heart, the inner membrane is 15 times larger. You see this, you can know the relationship between the mitochondria and the using of energy. Mitochondria has double membrane system. The outer membrane is freely permeable for molecule. In this chapter, in this lecture, you will not see much important about of outer membrane, but inner membrane has a lot to do for mitochondrial function. It has a protein complex. It has a protein that important for cellular respiration in the oxidative phosphorylation part. And in matrix, has an enzyme for citric acid cycle and has genetic system inside. I'm sorry, I did not tell you about the matrix. Matrix is here. The liquid part inside mitochondria. And the origin of mitochondria to the importance of the as I told you, after they found Krebs cycle, the scientists fascinated about the origin of mitochondria, and they spent 50 years studying. There is speculate that mitochondria is bacteria that engulfed by cells and live symbiosis and be symbiosis with the cells. And at that time, scientists cannot believe that something will happen. The bacteria inside our cells and live with us for like million years. Impossible. And they try to disprove this theory. But 50 years later, instead of disprove, they have to accept this theory that bacteria live with us. They call endosymbiosis. First, we have primitive eukaryote that still don't know how to use oxygen, but they already has nucleus when it's primary eukaryote. But at the time that we has blue green algae that produce oxygen, oxygen is abundant in the atmosphere. Some bacteria know how to use oxygen. It's an aerobic bacteria, and it's get maybe it golf or it can be, you can say it infect the primitive eukaryote cells. Eukaryote, this eukaryotic cell get advantage because the aerobic bacteria give them a lot of ATP from using oxygen, right? Happy to live together, and aerobic bacterium got a protection, so they stay inside for like 1,000,000,000 years. Live happily forever. And which kind of bacterium that is the ancestor of mitochondria? After like many 10 years and many years, scientists found that rickettsia may be the closest relative to mitochondria. Rickettsia can be related to Ehrlichia in dog. Rickettsia look like, this is a picture of rickettsia. And this rickettsia. It's similar to mitochondria by shape. There has a binary fission. Like so. Also intracellular living. Love to live inside cell. There has gene for respiratory change and ATP synthesis. Has circular DNA like mitochondria. And they can live like thousands in number in host cell, like mitochondria. And there already has a mitochondria. I don't know how to say it. Mitochondria DNA map related, very similar to that rickettsia. Too. So I think it's a proof that some kind of aerobic bacteria live with us, stay in our cell happily forever. The other thing is that mitochondria can change shape and size to meet a demand of energy. This is then from the old journal, but I think it's clearly show you. This is a picture of mouse mammary gland. The first one is when it's pregnant. You can see mitochondria look like this. Look at its size. Look at its color. And after give birth and lactating, the mouse need a lot of energy. Mammary gland need a lot of energy to produce milk. And look at its shape, size, and color. It's bigger in shape, and you can see it's got a lot darker because the inner membrane kind of dense inside. That's mean the cells change, the cell's metabolism change, and mitochondria change shape, change itself too, to meet the metabolic command of the cells. This picture shows that mitochondria has its own DNA. The point black here like this is a immune that was tagged to label DNA. You can see DNA that found near the membrane of mitochondria. Some eukaryote that love to be in the place that has that lack of DNA. You know, Giardia, Giardia that has a shape like this with two nucleus. That is the protozoa that live in and cause a disease in cattle. Giardia has two nucleus, only species that has two nucleus. They love to be in the place that has lack of oxygen. Some kind of eukaryote like this may lose their mitochondria. They may lose completely, or they still have remnant or some part of mitochondria. But because of their lack of oxygen in their environment, mitochondria is not need anymore. They just lost the mitochondria. But mostly us still have mitochondria. Mitochondria has their own genome, a circular DNA. They only has small genes, small amount of genes that produce rRNA, tRNA, and a protein for cellular respiration, the part of oxidative phosphorylation, which we talk later about this protein. Okay. Another things about mitochondria is about it can fuse together, or we call mitochondrial fusion. Two separated mitochondria. In this picture, two separated mitochondria. This one, the green one, and another mitochondria, this one, the red one, was labeled with the different color of dye. It can fuse together here, and the dye disperse into the another one, and the color mix. This one you can see. Turn into like orange. The benefit of fusion for the cells is that mitochondria can tolerate the high load of mitochondrial mutation. Like if the red one has a mutation, when it fuses to green one, the better the normal DNA can still working like that. Besides fusion to delay disorder, another thing about mitochondria, it can be fission for cell division. Here you can see this photo takes place in like 3 minutes. First, the green one is mitochondria. The red one is also mitochondria that was dyed in a different color. The green one, look at the green one first. They can be fission, separate 1 to 2. And a minute later, the second mitochondria can fuse to the red mitochondria. This one starting to fuse, and you see 3 minutes, around 3 minutes from the beginning of the take photos. It's turned orange. Fusion. They can fission and fusion. Fusion is to delay disorder, and fission is for cell division. We talk about structure, now we talk about functions. The mitochondrial function is to break down carbohydrate and fatty acid and oxidize them to ATP. When oxidation happens, electron was released, and in living cell, proton was released too. It's an exergonic. That means that release energy. Also, it can be heat too. It's important for heat production in the cells. And it's store of calcium in the mitochondria. This is a big part. I did not teach about this, but it's very important to know that there is high concentration of calcium in mitochondria. Mitochondria is like storage of calcium in the cells. And this one is important to induce apoptosis, which we will talk later, very later. Okay. Our second topic about mitochondria is about its oxidation and its ATP production. Okay. When you get sugar, like glucose with oxygen, we've got this carbon dioxide, water, and heat. To destroy one sugar, that will give you 2,000, almost 3,000 kilojoule per mole of sugar if you break it down. And when you break it down, this energy is trapped. Some of them turn into heat. Some of them will trap and use in cellular respiration. Cellular respiration has 4 steps: glycolysis, pyruvate oxidation, citric acid cycle, and oxidative phosphorylation. Let's see. Look at the overview. Here are the cells. The yellow one is cytosol. Here, cytosol. The big yellow here is mitochondria. First reaction, glycolysis, takes place in cytosol. It changes glucose into pyruvate. In glycolysis, or the older name is Emden-Meyerhof pathway. Glucose changed to pyruvate. On the way, they produce 1 ATP. And there is a oxidation. There will be electron released in here. And electron is not a good molecule, not a good particle to be near with. That has a high energy. There will be some kind of molecule that love electron and carry electron to another place. This is called electron carrier. In glycolysis, the molecule that carry electron is NADH, nicotinamide adenine dinucleotide. Carry electron in this NADH form. Pyruvate enter inner membrane, get into the second step called pyruvate oxidation. In pyruvate oxidation, will change pyruvate into acetyl-CoA. Acetyl-CoA. Let me see this. Pyruvate oxidation also give out electron and make one NADH. The third step is Krebs cycle or citric acid cycle. Citric acid cycle is important. It's in the matrix. Now in the matrix. And it's produced ATP and it's produced a lot of oxidation reaction, a lot of electron get out, and there is an electron carrier, NADH and FADH2. Electron carrier will carry electron that has energy inside. Move into the inner membrane of mitochondria into the last step, oxidative phosphorylation, and produce a lot of ATP. This is the overview of how ATP is produced. I will show you step by step, but not that much, not that in detail. But anyway, a lot of students don't like to know a lot of chemical name. I understand. It's too much. You don't have to remember it. But sometimes this reaction is one of the most important reaction in the cells. And you will heard about this name, that name many times. And if you know that what it is, that will be benefit for you for further study. Okay? You don't have to know very specific or you have to remember all of this. But if you know what it is, where it happened, and what is the product, and the importance of it in terms of normal work, a normal function of the cells or a disease, that will be helpful for you. First thing happen in cytosol, it's glycolysis. Glycolysis is to breaking down glucose into pyruvate. Glucose to pyruvate. And on the way, like this, there are eight or 9 steps. Glucose, and like this, and in some reaction, they release energy. They release electron that has energy, high energy enough for NADH to pick it up. And in some reaction, here and here, they didn't release electron, but they release high amount of energy itself. That ADP can couple this energy and make it ATP. Most of the time, ATP to ADP release energy for other people. But in this reaction, other reaction release energy, and ADP couple that energy and make ATP. This type of ATP making we call substrate level. Phosphorylation. So you can see that we can get ATP itself from glycolysis. Oh, sorry. Substrate level phosphorylation. Now here is one. I already talked. let's see, like conclusion. We degraded glucose. The degradation is the oxidation reaction, and oxidation is the loss of electron. In living cells, if you see loss of electron, you will see loss of proton together. And because electron is a very harmful particle, they need some kind of molecule that can carry electron. For example, NAD, when it's reduced with electrons, it turns into NADH. Like this, they call this kind of molecule electron carrier. Carry electron into another part of the cells. Electron carrier is a molecule that capable of accepting electrons from one molecule and then carry these electrons to another. Example that we will learn in this hour is nicotinamide adenine dinucleotide, NADH, or flavin adenine dinucleotide, or FADH2. NADH is nicotinic acid, which is vitamin B3. And flavin originated from vitamin B2. In making of ATP, we need a lot of vitamin. Mostly vitamin in the vitamin B group. You will see the importance of vitamin B5 and B1 later. So if you go to the drugstore and they say like vitamin for executive, vitamin for athletes, for working hard people, you will see this kind of group, vitamin B group, selling in the bottle to make your ATP making better. Okay, we finished glycolysis. Next, pyruvate oxidation. It's happened on the inner membrane. Before this, pyruvate oxidation is kind of in the cloud. They didn't pay much attention, but later, when they study this process, they know that pyruvate oxidation is like a door that open to the pyruvate to get into the mitochondria and make ATP. If the door is shut or the door is disabled, so no pyruvate can enter in, or less pyruvate enter into the mitochondria, and the cell will get less and less and less of ATP. Let's see, look at pyruvate oxidation. There is an enzyme that was like a door here on the inner membrane. This enzyme called PDC, or pyruvate dehydrogenase complex. This one. It's an enzyme that composed of 3 subunits of protein. The yellow one, the green one, and the pink one. It changed pyruvate into acetyl-CoA and release an NADH. Okay. And these 3 subunits is very important for scientists to study. Do you have do any student here come from Indonesia?
Yes, I am.
Indonesia. Yeah, you know this disease, beriberi.
Yes, Ajahn,
Ah,
very. When someone
beriberi.
lack of vitamin B.
You have a big institute there, right?
Yes, Ajahn.
ค่ะใช่the last 2 year student told me beriberi is a disease in Thai we call โรคเหน็บชา and the person who got beriberi what does it mean beriberi?
Beriberi is kind of disease when someone lack of vitamin B, as far I know.
I think I read in a textbook and they say that it's an Indonesian name, right? And beriberi mean I can't. Is it real? Is it true?
Pardon, I can.
Yeah, and because the person who got this disease, they cannot move, and it's kind of painful.
Yeah, some kind like that, Ajahn. The
And
people will feel the pain will show a weakness, loss of appetite because of lack of vitamin B.
yes, and it's vitamin B1, right? B1.
Yeah, I think so.
B1. It's happened when people start to not eating start to eat white rice, not the highly processed white rice, and it's scratch all the vitamin on the surface of the rice, which has a lot of vitamin B1. And this one, the complex, the first protein here, the yellow one, this one need vitamin B1 to work. If we are lacking of vitamin B1, this protein didn't working very well. So there will be less pyruvate to enter to the cells. And we will be less ATP. And that's why people feel weakness. Okay? Or in some type of toxin like arsenic. Arsenic will bind to this second subunit of pyruvate dehydrogenase complex. And when it bind to second subunit, this enzyme will less efficient, didn't working. That's why arsenic toxicity is like this. For example, when this enzyme has inhibitor, will be less pyruvate inside the cells. No pyruvate changing to acetyl-CoA. Oh, and if you look at acetyl-CoA, the structure of acetyl-CoA need vitamin B5 or pantothenic acid. Pantothenic, I'm not sure that how to spell it. Vitamin B5 up to pantothenic acid in its molecule. That's why I told you that vitamin B is very important for the making of ATP. You can see B1, B2, B3, and B5. And so in pyruvate oxidation, pyruvate will change to acetyl-CoA and enter into the matrix. Now we get enter to the third step of citric acid cycle of cellular respiration or citric acid cycle. You sometimes we call Krebs cycle. Acetyl-CoA combined with oxaloacetate make a citrate. That is why called citric acid cycle. And here, a cycle like this. On the way, there will be oxidation reaction and there will be electron loss. And electron loss can be catched by NADH. Sometimes it has a substrate level phosphorylation and make GTP. And GTP has an enzyme that can change GTP into ATP automatically without losing any energy. Or some reaction lost electron, but FADH come and catch the electron in ADH like this. Citric acid cycle. From one acetyl-CoA, you can get 3 NADH, 1 FADH2, and 1 ATP. I'm sure that you already know this reaction, the citric acid cycle, because the name is so short and it's a circular. It's easy to remember it in your picture in your brain. It's not only important like that. It's not only it's easy, but citric acid cycle and its glycolysis and its pyruvate oxidation produce is a major pathway, metabolic pathway. We can see this, we can found this reaction from very single cells, from the single cells, very common. And the intermediate in each pathway can be derived into another kind of molecule, maybe a nucleotide, maybe a lipid, a protein, an amino acid to make proteins. They provide intermediate to others pathway too. And in mitochondria, sometimes glucose, using glucose is not enough. Sometimes we have to burn fatty acid, burn our fat to make energy. That's process, beta oxidation. But the fatty acid was carried into the mitochondria, to the matrix, and change fatty acid into acetyl-CoA in ADH and FADH2 and enter citric acetyl-CoA enter citric acid cycle. I will not give you much detail, but don't forget beta oxidation. It's the second important way of using energy, way of give cell that energy. Now we talk about first, second, and third step: glycolysis, pyruvate oxidation, and Krebs cycle. Now, every electron that was released was captured by NADH or FADH2 and enter into inner membrane of oxidative phosphorylation of mitochondria into the fourth step, oxidative phosphorylation. A little different here is that NADH from cytosol enter mitochondria. They have to pass through inner membrane here, which is permeable membrane. In some cells like heart, brain, NADH from cytosol may enter the cells, may enter inner membrane, enter into oxidative phosphorylation in the term of FADH2. A little bit strange. In some cells, like liver, NADH enter to oxidative phosphorylation in the NADH form. But in heart or brain, it's enter oxidative phosphorylation in terms of FADH2, which is, you can see that it's stupid, but actually, with this kind of process, the cells the pyruvate can even very low concentration. Even when it's very low concentration of food, it still can enter the cells, enter the mitochondria. Now let's see oxidative phosphorylation. I love its name very much. Its name is oxidative and phosphorylation. There are two mechanisms, two steps very clearly. Oxidation on the right and phosphorylation. Oh, sorry, I always confuse. Oxidation. And the second step is phosphorylation. In oxidation, there is a release of electron and electron moving along the way. It's called electron transport or electron change. Phosphorylation is adding of phosphate to ATP and make it ADDOC and make it ATP. The mechanism here we call chemiosmotic coupling, which we will see later. And I will show you oxidative phosphorylation. Here is inner membrane. Here is mitochondria. We are talking about oxidative phosphorylation in the mitochondria. This is inner membrane of mitochondria. This is matrix. The outer membrane is here. And here is intermembrane space. Okay, I will get rid of. And on inner membrane, there will be a protein, enzyme, floating in the inner membrane. It's a respiratory enzyme complex. It's composed of protein 1, 2, 3, and 4. I didn't mention the name and some kind of lipid like coenzyme Q. Or cytochrome C. Here. Now let's follow the electron, right? Follow the electron, follow the energy in the electron. In oxidation steps, NADH carry electron and send electron to protein complex 1. May I change the color? Let's change the color. Electron from NADH was sent to protein complex 1. An electron moving in complex 1, an electron was sent to coenzyme Q. Coenzyme Q bring electron to complex 3 and send electron to cytochrome C. Cytochrome C send electron to complex 4. At last, oxygen come and bring and receive electron and change into water. The final product is water here. The electron end up into water. Oh. Okay. Electron is moving along this protein. This protein. And oxygen receive that electron and change into water. That's the end of dangerous particle, high energy particle. Along the way, when electron pass the protein complex 1, 3, and 4, proton will be pushed outside matrix. Here. Here. And here. When electron pass, proton was pushed into the intermembrane space. Finally, the pH of the intermembrane space is different from in the matrix, like 1.4 pH. The difference in pH, the difference in proton concentration make a potential, different potential. And when the concentration is different, the thing that happen is that the osmosis occur. The proton try to move into the matrix. Okay, try to move into the matrix. They move past through the fifth protein. That is, the name is ATP synthase. ATP synthase. The proton moves through ATP synthase and cause ATP synthase to change their structure. The changing structure cause the energy coupling AD-P to ATP. That's why they call this step the phosphorylation here. The mechanism is chemi-osmotic coupling. Come from osmosis, chemi-osmosis. Here and the coupling here. Okay, let me delete something. And because NADH release 3 proton, they believe that one NADH can make 3 ATP. And because FADH pass only 2, so they believe that one FADH give 2 ATP. But if you read newer textbook, they will told you that one NADH give you only 1.5 ATP and one FADH2 give you 1.5 ATP. But however, whatever it is, you understand the mechanism, and that is all. Okay, oxidative phosphorylation, oxidation in electron transport. Electron in NADH and FADH2 is transport to oxygen and make water, right? Electrons are transferred through a series of electron carrier 1, 2, 3, 4 and organized onto 1, 2 protein complex. Okay. The phosphorylation steps, chemi-osmotic coupling. Because there is a generation of electrochemical gradient. This is inner membrane, right? Proton out. This is a proton here, higher. So it's called electron chemical gradient. Proton want to diffuse back, called chemi-osmosis. When it's come back, energy was coupled by AD-P and make ATP in ATP synthase. Happen in enzyme called ATP synthase. So they call chemi-osmosis, chemi-osmotic coupling. ATP synthase look like this, so cute, look like a doorknob. If you are but it's easier for Asian, we look it like it's a mangosteen. And if you look in the electron microscope in the mitochondria, this is a matrix of mitochondria. And inner membrane here, you can see the doorknob, ATP synthase. There are one of the high study protein. I'm very bad at maths, but I think some of scientists love the kind of mathematics, kind of engineering, engineer. And they look at ATP synthase like something mechanical and try to study it in terms of engineer, to study how it works and try to simulate this kind of structure and make it kind of like bio-robot, something like that. I don't understand the how can they make it, but I understand the foundation of not foundation, why they love to study it. It's fascinated. The structure very complicated, you can see. This one proton get into this one, I think here, and proton that enter it, the energy in it change the structure of this, and ADP enter it, get out as ATP. You can watch YouTube. Lately there are a lot of YouTube that simulate cellular respiration, ATP synthase, this kind of all, but not complicated. There are many YouTube that you can watch cellular respiration and ATP synthase. Some may look like I told you, some may not look like, or some may very much in detail. Whatever it is, you can watch it and kind of memorize it a little, understand what happened. It will help you a lot when you have to use your no. If you have to use it like a quick wit, like a quick understanding, okay. And this is theoretical yield. Many teachers love to ask students, like, 1 molecule of glucose, how many ATP that you can make, like that. So I just like give you a summary, okay. Glucose, first step is glycolysis. To degrade glucose, first step is glycolysis. There has ATP by substrate level phosphorylation. And the second step, pyruvate. give to NADH, and NADH can enter intermembrane, can enter inner membrane, and depend on cells. Some cell it's enter mitochondria like NADH, it gives 6 ATP, but in some cells it's not, give 4 ATP. Pyruvate oxidation, pyruvate change into acetyl-CoA as 2 NADH, so it gives you 6 ATP. Acetyl-CoA enters Krebs cycle. Oh, I'm sorry, I have to tell you that this glucose to pyruvate, one glucose give you 2 pyruvate, so it will be like this. So Krebs cycle, in Krebs cycle, one Krebs cycle give you 3 NADH, so 2 Krebs cycles give 6, give 2 FADH, and give 2 ATP. Actually, GTP and change to ATP. Like this, and total net protein yield is 36 to 38. Depend on what kind of cells. And if you're looking at the newer textbook, it's gonna be like 30 to 32 ATP. I don't mind this. It will change on time, I think. Why I say that? Because if you study all of this, you need to study by in vitro, in vivo, no, not in the real situation. In the real situation, we don't know what exactly it is in the cells, right? Mostly we study in scientists study at this kind of temperature, at the ความดัน pressure. What pressure inside the cell we don't know. The concentration of the substrate we really don't know. It's not the real concentration. Here or here is estimated value. So after ATP is produced, ATP will export out of the mitochondria by adenine nucleotide translocator. It's another enzyme in exchange of ADDOP. And in bacteria also has cellular respiration. The origin of mitochondria. Bacteria has used cell wall instead of outer membrane, and the cell membrane is used instead of inner membrane. And oxidative phosphorylation can be happen in bacteria without having a mitochondria. And I have to tell you this. I'm sorry. If without oxygen, the anaerobic respiration, one glucose give only 2 ATP. With oxidation, with oxygen, 18 or 19 times higher in ATP production per glucose. So a lot more of by using oxygen, oxidative phosphorylation that give a higher yield of ATP and allow this higher yield allow cell to do a lot more work, right? This is bacteria. No, sorry. This is bacteria. Some living organism that didn't use oxidative phosphorylation still be unicellular living organism, but the organism that use oxidative phosphorylation can produce more ATP, and cell has a lot more function, highly developed, a lot evolution happen in it. The cell can be developed or evolution to like nerve cells, to movement, or to as active in like immune cell, like that. That's the importance of oxidative phosphorylation. And now we know that how mitochondria work, how oxidative phosphorylation, cellular respiration happen. Now what's happen if that we talked before is not happen? No, is abnormal. This kind of area, mitochondrial disease, is I think like 20 was put on the table like 20 years ago, and it's still now it's taking more control, more important, and people study a lot. There will be a group of disease that was categorized into mitochondrial disease. Now, mitochondrial disease is the disorder that result from the dysfunction of ATP production. Cell will be in crisis. The ATP is produced not enough for the need. They can be caused by a mutation, either nuclear DNA or mitochondrial DNA. Because when bacteria come into the cells and symbiosis, it's delete some of the genome that can be used with the host cell. Or look at the other side, is that the host cell try to take control of the bacteria, so they send some enzyme that they can produce and send it to the bacteria to use, so they can be live happily forever, like that. So when mitochondrial disease happen, when the nuclear DNA mutation or mitochondrial DNA mutation can also cause mitochondrial disease. The result is the less of ATP, and the other result is that more oxidative stress. Why oxidative stress? In oxidative part, there is electron transport, right? Normal electron can be leaked from the electron transport, but if in mitochondrial disease, we found that electron is leakage from electron transport a lot more, and Electron is a high energy particle. Electron can attack lipid, attack protein and DNA, and cause a changing in that molecule structure. One of the things that you can see it in your daily life. You know this molecule, right? We call phospholipid. It's fatty acid. Two legs here is fatty acid. And one leg is king. That makes phospholipid is difficult to attack to each other. And make fluidity on the cell membrane. That makes cell membrane move in the plane of it, because one of the fatty acid leg is kink. What happens if electron attack to fatty acid here? Electron make the kink here like this. And fatty acid can be a much nearer. Which make plasticity on the cell membrane is less fluidity. Or if they attack the protein, maybe enzyme protein something, that make that protein less efficient to do work. Or if they attack, the electron attack DNA, can cause DNA damage, cause mutation. And maybe it's not happen in one single incident. It just accumulated. It just happened a little bit, but every day, it will accumulate the dysfunctional molecule. Besides less energy, the cell can slowly degrade it or make tumor or aging. And if lipid oxidative stress still happen and more, it causes mitochondria to swell and cytochrome C and calcium release and induce cells to death or apoptosis. This is the picture just to show that in mitochondria, in the inner membrane, see, there is electron transport here. Electron transport here. Electron can leak out and catch with oxygen. When electron and oxygen combining together, they cause different kind of molecule, like a species. So they call this one like reactive oxygen species. This. That has electron inside and can attack protein, lipid, and DNA, like I said before. It's not all bad things. In some cells, we use reactive oxygen species like a signal transduction, like to tell the cell to do something. Like, bacteria inside is invaded or virus invaded, we have to kill it. We use this kind of signal and use this reactive oxygen species to kill the invader. But if it's a lot of oxidative stress, it's not good for the cells. You will find this regularly in your study, reactive oxygen species. Now it's like the cause of almost everything that make the cells disable, make the cell abnormal. And this one, reactive oxygen species. And if you are a girl, you will see a lot of cosmetic product that said or advertised like antioxidant. Or you eat something like highly antioxidant. That's anti-reactive oxygen species that cause cell damage or cause aging. Okay. So, oh, sorry, it's 3 p.m. Oxidative phosphorylation in metabolism can cause oxidative reaction. Electron can leak and make reactive oxygen species. If it's not much, it can trigger a cells to signal to do something or to defend cells. But if it's too much, it can damage cells. Tissue dysfunction and aging. It can cause oncogenesis and cause cancer. Metabolic mitochondrial disease. A student asked me if it's mitochondrial disease in animal. I can say that it's we didn't think about it much because mitochondrial disease, some animal, if it happened, they're still born, they die before it's born, or they die when it was young, like that. But there are some cases that recorded about mitochondrial disease, like this in dog, German Shepherd dog, exercise intolerance and atrophy of muscle. When it's die, when the dog die, they measure they found that cytochrome C oxidase activity, which is the one of the 4 protein on oxidative step in oxidative phosphorylation, is absent. They found a typical mitochondria structure like this. Or in a family of golden retriever, they found sensory ataxis neuropathy. It's abnormal. It linked to mitochondrial disease. And mitochondrial disease, now they know some method to correct mitochondrial disease. If you read the news, like I think 4 years ago, there is 3 biological parents. There is a woman that has mutation of mitochondrial DNA. She herself can overcome the disease, but I think she miscarried. She lost her baby before it was born, like 2 or 3 times, and they found that she has mitochondrial disease, that the embryo cannot be born like that. So they bring out her genome, nuclear genome, and there are another egg from another woman with normal mitochondria. act as a second parent. They remove her genome, let alone the mitochondria. Only mitochondria of the second woman left in the oocyte, and they combine 1 and 2 together. They make the IVF, cause the new baby. It's happened, and the baby still alive and well, and nothing abnormal about them. So I think it's time. So now in the mitochondria, there has a function in our cell. For life, they produce energy for us. And they also produce something that induce the cell to die. Life and death by mitochondria. Okay. I think we meet again in apoptosis topic. Okay. So this is the end. I'm sorry, I take you like 5 minutes more. About the examination, we will not see each other. I will give you a question, and you do the research. I will put it in the line, the group line of us. Okay, and you don't have to write down a lot. I think maybe 4 and 2/3 of the page is enough. If half pages is too little, 1 pages is too much. I think 2/3 of the page is fine for me. I will give you a question and you write down to me. Okay? Give you an assignment instead of the exam. Give you in the line. Okay. If you have any difficulties, you can write it down into the in the line group. I will read it and your friend will know what is the problem. Okay. I'm sorry that we cannot see each other. Really sorry. But if you have any question, and you come to the faculties, you can ask me, or you can ask me in line group, and it's maybe difficult for me to write, to type it down for you, but I will try. Okay, if you do not have any questions, so you can take a break. Okay, so now bye. Everybody has a take care of yourself. Okay.
Okay, adjourn. Thank you.
Thank you
ค่ะ
ขอบคุณค่ะ
สวัสดีค่ะ สวัสดีค่ะ
Take care, John. Goodbye.
Bye